Hello students, welcome to EPG Pathshala. I am Professor Praveer Kumar from Department of Biotechnology, Delhi Technological University. Today we are going to discuss about a module understanding the architecture of proteins and protein to proteome under genetic engineering paper and recombinant DNA technology. So, my talk will be divided into few parts, starting from introduction to protein, then protein structure and amino acids, protein structure and its folding, and proteome and proteomics. So you can see that upon translation, when protein or polypeptide chains are produced, it has different structure. It has the linear and primary structure, secondary structure, tertiary structure, and quaternary structure. And the various functions of protein is in different regulation part, signaling part, transportation part, movement part, structural components, and many more. So protein, you can say that also a building block of humans or any cell. Protein has various functions, as I discussed, that in signaling, because the protein molecules can help in interacting between two molecules or between two proteins. There are many transporter proteins which facilitate the work. Regulatory part is also very important in under protein function. And of course, there are many structural tissues which have been made up of protein. Protein is also important for the movement part, and let's see once again repeating central dogma of life. So starting from DNA, it is transcribed and the protein to mRNA, and mRNA is translated to protein, and this protein has the 3D structure once it's formed and fall into the cytoplasm, where it has getting the 3D structure which is modified under different chemical and chemical modifications, and then it got a new 3D structure, and thus it performing a function or a new function. Amino acids are compounds made up of carbon, oxygen, hydrogen, and nitrogen. They act as monomers of proteins and are consisting of carboxyl group, an amino group, a hydrogen atom, and a different side chain, all bonded to the alpha carbon. In an alpha amino acid, the amino and carboxylate group are bonded to alpha carbon. Further, various amino acids differ in respect of the side chain, that is the R group, attached to their alpha carbon. And you can see here the common structure of an amino acid. There are 20 amino acids which is linked through the peptide bonds. And in any protein, polypeptide is the backbone, where the R group is, which R group or side chain is present. The structure is common to all except one of the alpha amino acid, and that is proline. The R group of side chain attached to alpha carbon is different in each amino acid. In the simplest case, the R group is a hydrogen atom and amino acid is glycine. In alpha amino acids, both the carboxyl and amino group are bonded to the same carbon atom, although many naturally occurring amino acids not found in protein and have a structure that differ from alpha amino acids. In these compounds, the amino group is attached to a C carbon other than alpha carbon atom, and they are called as beta, gamma, delta, epsilon amino acid depending upon the location of the carbon atom to which amino group is attached. As I said you, there are 20 amino acids which is broadly divided into polar amino acid and non-polar amino acid, which means hydrophilic part is the polar group and hydrophobic part is non-polar group. However, there are more than 300 amino acids are present in the cell, but only 22 amino acids take their part in protein biosynthesis ribosomically. Such amino acids are known as standard or proteinogenic amino acids. Some amino acids are more abundant in protein than other amino acids. 4 amino acids, serine, lysine, leucine, glutamic acids are most abundant amino acid residues in a typical protein, whereas tryptophan and methionine are rare amino acids in a protein. Standard L-alpha amino acids are specified by simple 3 letters codon, and an amino acid can be classified on the properties of their side chain. The polarity of the side chain varies widely from non-polar and hydrophobic, as discussed, to highly polar and hydrophilic part. Amino acid with non-polar side chain. Here, among standard amino acid, 9 amino acids contain non-polar side chain that includes alanine, glycine, leucine, valine, proline, isoleucine, phenylalanine, methionine, and tryptophan. Proline differs from other members because of having its side chain bonded to both the nitrogen and alpha carbon atoms, whereas tryptophan and phenylalanine are having aromatic side chain. Further, the side chain of tryptophan contain an indole rings, whereas phenylalanine has a phenyl ring. Come to the second part, that is the amino acid with uncharged polar side chain. Here, there are 6 amino acids contain uncharged polar side chain. These are threonine, cysteine, serine, asparagines, tyrosine, and glutamine. 3 amino acids, threonine, serine, and tyrosine, are having hydroxyl group attached to a side chain, whereas cysteine is similar to serine but contain a thiol or sulfahydryl group in place of OH group. Come to the third part, that is the amino acid with charged polar side chain. Here, the positively charged R group, arginine and lysine, have side chain that contains positively charged group at neutral pH. Lysine has an amino group, whilst arginine containing a guanidinium group, histidine contains an imidazole group and aromatic ring. Now, come to the protein structural organization. So, proteins are unbranched polymers constructed from 22 standard alpha amino acids. They have 4 levels of the structural organization, and that is primary structure. The amino acid is specified by genetic information. As the polypeptide chain folds, it forms certain localized arrangement of adjacent amino acids that constitute secondary structure. The overall 3D shape of a polypeptide assumes or known as tertiary structure of a protein, and likewise proteins that contain 2 or more polypeptide subunits are said to have quaternary structure. In the protein structure, there are several chemical or other kind of modification which is important to perform a function. For example, acetylation, phosphorylation, hydroxylation, methylation, carboxylation, glycosylation, ubiquitination, and you can see it here in the right hand panel that these are the branches where the modification or chemical modification occur, and thus the protein has performed a special function for the cellular activity. So the proteins are non-covalently bonded, and you can see it here that in this, here there are 3 structures, and you can see that the proteins are either covalently bonded through ionic bond or hydrogen bond, and you can also see that how a van der Waals attractions is playing role to give a proper shape of protein. Again, in the hydrogen bonds in proteins, there are three different things you can see: that hydrogen bonds between atoms of two peptide bonds. Second, hydrogen bond between atoms of a peptide bond and an amino acid side chain, whereas the third one is the hydrogen bond between two amino acid side chains. So in between and inside chain, how hydrogen bonding is playing an important and crucial role, you can see it there. Now come to the primary structure of protein. The primary structure of a polypeptide is its amino acid sequence. The amino acids are connected via peptide bonds. Polypeptide containing primary structure that determines the higher level of structural organization, as you can see in the figure as well, that how alpha helix and beta strands are traveling through the linear structure of amino acid. Come to the secondary structure. The most common type of secondary structure are the alpha helix and the beta plated sheet. Both alpha helix and beta plated sheet patterns are stabilized by hydrogen bond between the carbonyl and NH group in the polypeptide backbone. The alpha helix is a rigid, rod-like structure that forms when polypeptide chain twists into a helical conformation. The screw sense of alpha helix can be right-handed or left-handed. However, right-handed helices are energetically more favorable, and length of alpha helix is usually 10 to 15 amino acid residues. Interchain hydrogen bonds form between the NH group of each amino acid, and the carbonyl group of the amino acid has 4 residues away. Further, beta plated sheet form when 2 or more polypeptide chains segments line up side by side. Each individual segment is referred to as a beta strand. Now, come to the tertiary structure of a protein, where the term tertiary structure comes from or referred to the unique three-dimensional conformations that globular proteins assume as a result of the interaction between the side chain in the primary structure. All the informations required to fold protein into its native tertiary structure is contained within the primary structure of the peptide chain itself. The following types of covalent and non-covalent interaction stabilize tertiary structure of a protein, and these are the non-covalent interaction, hydrogen bonding, electrostatic interactions, hydrophobic interactions, van der Waals force of interaction, where the covalent, as I discussed in the previous slide, the most prominent covalent bonds in tertiary structure that are disulfide bonds present in several extracellular proteins. Cysteine has a thiol group that is unique among the 20 amino acids in that it often forms a disulfide bond to another cysteine residues through the oxidation process of their thiol groups. Now come to the quaternary structure of protein. As I discussed in the primary, secondary and tertiary, and the quaternary structure has numerous protein such as hemoglobin, which is a complex protein, and it is made up of 2 or more polypeptide chain. Each polypeptide chain is known as subunit. Subunits in multimeric protein may be identical or quite different. Polypeptide subunits assemble to form quaternary structure and are stabilized by non-covalent bond, including hydrophobic interaction, electrostatic interactions, and hydrogen bonds, as well as covalent cross linkers. So if we summarize the previous slides, we have learned that peptide bond rotation and R group orientation is very important in protein folding. Not only that, weak non-covalent interaction is also required for a proper shape of protein. Protein shape and sequence of amino acid is also important because it can also help in the conformation and determining the free energy, whereas denaturation and renaturation of protein is a continuous process, and there are many factors such as heat, pH, chemicals and times are important in protein folding. To begin how protein folding work in the cellular milieu, I just want to give you some information that inside the body there are about 70% proteins are folded properly, whereas about 30% proteins are prepared or formed without being properly folded. So cellular machinery is trying to give that non-folded or unfolded protein to a proper shape, and thus what is happening, the protein which is unfolded or aggregated or misfolded in nature has to move through a certain machinery which is called the chaperonic machinery. And here what is happening, that these proteins are going through the chaperonic machinery, and chaperone is trying to give a proper shape of that misfolded protein. However, there is an issue here that all non-folded, misfolded or aggregated proteins are not properly folded, and in that situation what is happening, the cell is trying to eliminate those proteins which is not been guided or folded by chaperones, and thus there is a machinery called ubiquitin proteasome system is playing in action, and it can release or it can remove the non-folded proteins through the system. Now the question arises here that which protein should be inside the cell and which protein should be discarded can exclusively be decided by an important protein and cognates which is called the ubiquitin E3 ligase protein, because ubiquitin E3 ligase enzyme is the decisive factor for any protein that has to be discarded from the cells. Now here you can see that a primary folded or misfolded proteins came out from the ribosome, and it can enter into the chaperonic system, and you can see here that protein is traveled to that chaperonin with the help of the ATP and capping. This protein is trying to got a proper shape, and thus once this capping is removed, the properly folded proteins comes out. We all know about the pathophysiology and progression of Alzheimer's disease, where amyloid beta is aggregated and making plaque. So what is happening here, that in every individual cells there is a A-beta monomer, and these A-beta monomer, you can say it in the precursor form and is non-lethal, that this monomer of A-beta is not making or not doing any harm in the body, but due to some signal change or oxidative stress or any environmental insults, these monomer form is getting or becoming oligomer, and finally this oligomeric pattern of protein is converted into fibril, and ultimately the signal between the two synapses is restricted. Here I am giving you just an example of the misfolding protein, because here the proteins, non-functional proteins are aggregated, and thus here we can say that how a plaque is formed, and between the two synapses or between the two neurons communication got disturbed, or there will be no more communication. And you can understand now why do we need protein folding, because all the misfolded, non-folded or not properly folded protein can cause a detrimental effect on human body or cell per se. So here you can see that the purified protein isolated from cells. Now I am going to discuss about the in vitro situation, that how you can say that you can play or you can manipulate the protein structure. So you can see it in this figure that purified protein isolated from the cells and exposed to a high concentration of urea. The moment you added the urea, that protein is denatured. Remove the urea and again that protein is got their original conformation. So here we have learned the denaturation and renaturation process of protein. Now come to the different protein domains, because this is also important in the signaling. Not only the signaling, there are many cellular processes where the domain structure of a particular protein is playing crucial and decisive role. So domain is a basic structure where protein can fold into a stable structure independently. Different domains impart different function, and protein can have one to many domains depending on the size, and this I can show you in my next slide, that what are the importance of various domains and protein structure. Here you can see adapter protein, scaffolding protein, kinases, phosphatases, RAS signaling molecule, transcription molecule, ubiquitination, and many more. So you can see in all this list there are many proteins which have many boxes, and these boxes are basically domain which playing crucial and decisive role in a cellular function. Now I am going to discuss about the post-translational modification. As you know that these processes are very important to perform many protein functions. As you see in the domain structure, there are many boxes, and I have already said that post-translational modification is very important to give a particular domain or a particular amino acid or a particular chemical to perform a selective function. There are many post-translational modifications such as acetylation. So I will discuss the entire process one by one. In the acetylation, where an acetyl functional group into a chemical compound get introduced. Conversely, deacetylation is a process of the removal of an acetyl group. Acetylation refers to the process of introducing an acyl group resulting in an acetoxy group into a compound, namely the substitution of an acetyl group of an active hydrogen atom. A reaction involves the substitution of hydrogen atom of a hydroxyl with an acetyl group yield a specific ester, and which is called acetate. Acetic anhydride is commonly used as an acetylating agent reaction with free hydroxyl group. For example, this acetylation process is used in the synthesis of aspirin, heroin, and THCO acetate. The another important post-translational modification of a protein is mediated through phosphorylation, and phosphorylation always coupling an addition of a phosphoryl group. This particular biochemistry probably represents the origins of catecholamine overloaded to a molecule. In biology, phosphorylation and its counterpart, dephosphorylation, are critical. For many cellular processes, a large fraction of proteins are temporarily phosphorylated, as are many sugars, lipid, and other molecules. Phosphorylation is specifically important for protein function, as this modification activates or deactivates almost half of the enzymes, thereby regulating their function. Protein phosphorylation is considered the most abundant post-translational modification in eukaryotes. Now come to the third post-translational modification process, that is called the hydroxylation. Hydroxylation is a chemical process through which introducing of a hydroxyl group into an organic compound. In biochemistry, hydroxylation reactions are often facilitated by enzymes called hydroxylases. Further, the process of hydroxylation is crucial for oxidative degradation of organic compounds in air. Moreover, it is also pivotal in detoxification process as hydroxylation converts lipophilic compounds into hydrophilic products that are more readily removed by the kidneys or liver as excreta. Some drugs, including steroids, are activated or deactivated by hydroxylation process. In this series, the another important post-translational modification is DNA methylation, which is a very important process in the biochemistry where methyl groups are added to the DNA molecule. The process of methylation is responsible for altering the activity of a DNA segment without affecting the sequence. Further, it acts to repress gene transcription when it is located in a gene promoter. It plays a decisive role in normal development and found to be associated with numerous key processes such as X chromosome inactivation, genome imprinting, carcinogenesis, repression of transposable elements, and aging. Now come to the carboxylation process. In this process, carboxylation in the biochemistry is a post-translational modification of glutamate residue to gamma carboxyglutamate in proteins. It occurs primarily in proteins involved in the blood clotting cascades. This modification is required for these proteins to function. Carboxylation occurs in the liver and is performed by gamma glutamyl carboxylase. The carboxylase requires vitamin K as a cofactor and performs the reaction in a processive manner. Gamma carboxyglutamate binds calcium, which is essential for its activity. For example, in prothrombin, calcium binding allows the protein to associate with the plasma membrane in platelets, bringing into close proximity with the proteins that cleave prothrombin to active thrombin after injury, and this is only an example that how prothrombin is converted to thrombin and how the blood clotting cascade is completed. Now come to the glycosylation process. Glycosylation is a form of co-translation and post-translation modification. In biochemistry, glycosylation refers in particular to the enzymatic cascades that attaches glycans to protein, lipid, or other organic molecules. This enzymatic cascades generates one of the fundamental biopolymers found in the cells. Further, in this process, a carbohydrate, that is a glycosyl donor, is attached to a hydroxyl or other functional group of a glycosyl acceptor. The majority of protein formed in the RER, that is the rough endoplasmic reticulum, undergo the process of glycosylation. It is an enzyme-directed site-specific process which is present in the cytoplasm and nucleus as the O-glyconation, or also called O-linked glycosylation modification. There are 5 classes of glycans are produced, that is N-linked glycosylation, O-linked glycosylation, phosphoglycan, C-linked glycan, and finally glycation. Here you can see N-linked glycans bonded to nitrogen of asparagine side chain. N-linked glycosylation requires participation of a special lipid called dolicol phosphate, whereas O-linked glycans attach to the hydroxyl oxygen of serine, tyrosine, threonine, and hydroxyproline side chains or to oxygens on lipids such as ceramide. Phosphoglycans attach to the phosphate of a phosphoserine, whereas C-linked glycan, a rare form of glycosylation where a sugar is added to a carbon on a tryptophan side chain. Finally, glycation, which is the addition of a GPI anchor like protein that links protein to lipid through glycan linkage. Come to the next modification, which is also a very important modification through which the misfolded or non-wanted proteins are discarded from the cells, and that process is called ubiquitination. The addition of ubiquitin of a substrate protein is called ubiquitination, or less frequently ubiquitination. In another word, you can say the destruction of a protein is called ubiquitination. Ubiquitination affects protein in many ways. It can mark them for the degradation through proteasome, impair their cellular location and activity, and finally prevent protein interactions. It basically involves three crucial steps, that is activation, conjugation, and ligation, through which an orchestrated E1, E2, and E3 enzymes are playing a crucial role. The outcome of this chronological cascade is to bind ubiquitin to lysine residues on the protein substrate via an isopeptide bond, cysteine residues through the thiester bond, serine and threonine residues through an ester bond or N-terminus of the protein via a peptide bond. So to conclude my talk today, what we have learned that protein is very important to maintain cellular structure, architecture, as well as function. So now one can ask, what would be the end goal of proteomics? So the application of proteomics is important or meant for protein mining, protein expression profiling, protein network mapping that I have already said you in the signaling part, and mapping of protein modification. So these are the important things which is achieved or governed through the proteomics. So if again we can recapitulate the central dogma of life, the transcription, translation, and enzymatic reaction is achieve our goal, that is from DNA to RNA and RNA to protein and protein to metabolites, and how these metabolites are comes under action in the cellular machinery. So you can see here genomics, transcriptomics, proteomics, and metabolomics, and if you add the transcriptomics and proteomics, it will come under the head of functional genomics. Thanks for your attention.